data from the 8P heroes and really trying to understand baselines and also start to ask some questions about what's really going on in this disease, because this is actually, after all, a new disease to medicine. We take it for granted. We think about 8P all the time, but the rest of the world, doesn't. And the fact is, there's a lot of terra incognita we still need to explore. So we'll have some clinical experts talk to us about similar efforts that have been underway in other communities, other neurodevelopmental disease communities. So really excited to tell you more about the Health Canvas study. Again, there'll be a Substack post that I'm putting out. We'll also have next month's roundtable devoted to exploring what the Health Canvas study is all about. And then a little bit on the science side. So we're excited. Some of the folks who have given talks at this roundtable will have a chance to get on stage, especially some of the younger scientists who've been driving the work at the bench. So they're going to have a chance to talk about some of our research highlights in chromosome engineering and disease modeling. So really excited about that. We'll have our friends from Integrated Bioscience join as well. We'll talk about the integrated stress response and our first real attempt at understanding potential therapeutic mechanisms that might be beneficial for 8P. And then we're going to have some breakout sessions, some working groups. We're going to have some brainstorms. We're going to talk about the convergences that we're seeing between 8P and other neurodevelopmental chromosome disorders. We're going to talk about some of our favorite driver genes. We'll try to also stimulate discussion around drivers that we may not have paid attention to before. And you can check out the latest Substack about driver genes to kind of wet your whistle and get ready for that. And then we really also want to take advantage of having all these great minds in the same room, and we don't want to just have the energy of the conference dissipate after we leave Denver. So we really want to focus on grant opportunities and how we can put together multi-PI grants among our researchers, and just obviously also help facilitate our AP researchers where we can to help them leverage the data that has been generated with Project AP funding and get now grant support, so you can grow your lab and grow your commitment to AP. So one final note about the conference: we're also going to have a medical fair where we'll be able to hopefully have some cool technology. It's all exploratory, safe, but we'll have some cool technology for neuro assessment and cognitive assessment and other measures that we hope will also teach us some things that, again, will ultimately get us ready for clinical trials. Anything else, Bina or Barbara, you want to add to the conference preview here? If not, then really would like to have our guest today from a company called Spring join us. Maybe Ben Kamens, the CEO of said Spring, can maybe introduce himself and the company, and then hand it off, I think, to a teammate who will walk us through their technology and a case study, and then how their technology can specifically help and focus on AP.
Absolutely. And hey, we're really looking forward to this and grateful to be presenting to this group. So thank you all for your time very much. We will walk you through both our company and our case study and how we think it might apply to some of the research that you're interested in. But maybe just start with really quick intros. I'm Ben. I'm the founder and CEO of Spring. Rachel, I'll hand it to you briefly.
Hi, I'm Rachel. I'm head of R&D at Spring. Tim.
Hi, Tim. I lead all business development and corporate development at Spring.
And Adi.
Hi, I'm Adi. I'm a machine learning scientist.
And so what we're going to do is share quite a few different things with you. I'll go ahead and just briefly share my screen. And I assume you're able to see this? Great. Rachel, do you want to take us through the agenda for the presentation, and then I'll give a high-level overview?
Sure. Yeah. So Ben's going to give you the high-level overview, so an introduction about our company, how we work together as a team and with other groups to address really complex biological questions. And then I'll take back over. I'm going to walk you through kind of at a high level how we use cell painting to generate data that we then use to train AI tooling to address complex biological questions, and then get into a case study of a phenotypic screening effort that we did, where we combined AI tooling and cell biological features, known readouts, to identify compounds that were functionally diverse in the same phenotypic screen. So we were looking for different mechanistic compounds and seeing how we could classify those to match, optimize to patient needs. And then lastly, we'll end on a proposal that we've put together to identify therapeutics for 8P heroes, and how we can use all of our experiences in drug discovery and AI to surface effective therapeutics. So I will pass back to Ben.
Yeah.
So, just before you get going, Ben, here, so I just want to just comment that we have a diverse audience here of researchers and families, and so we have professionally trained scientists and budding scientists. But if we can possibly kind of be as jargon free as possible, I think that just keeping that in mind, and then I may take a liberty here or there to jump in, maybe just to have a point clarified or go a little deeper if I feel like maybe this is a point that the aficionados get, but maybe we should dwell a little longer for everyone else. Anyway, sorry, cut you off and jump back in.
Absolutely, and that sounds good. And we are a team that, as I'm about to show you, comes from quite diverse and different backgrounds, some from drug discovery, some from the computational world, and we are used to having to learn each other's languages and ask questions about it. So certainly jump in if we're off target in any way there with our presentation. Just to emphasize, so we're a small company, we're about 30 people, and our whole goal is to put new technology tools in the hands of research groups and in the hands of scientists so that they have all the latest and greatest superpowers that new computational approaches like the world of artificial intelligence can give to those who are doing drug discovery and development. That's our whole mission. And we tend to work with a group that's very, as I mentioned, cross-domain. Whenever we work with partners, we're always putting together teams of research leaders like Rachel on the call here, our R&D leader, folks from a machine learning or AI background, folks from a software engineering background, folks from a classical biotech background, and we put those groups together in order to service the folks who we're partnering with. And so that cross-domain focus, you'll see both in our the composition even of this meeting of who we brought, but also throughout the work whenever we work with somebody. You don't need to read everything on this screen. Just to focus on the right side for a second, I just want to emphasize we do work with big pharma companies, we do work with small biotechs, we do work with nonprofit research groups like Bill and Melinda Gates Foundation, academic groups like the Broad Institute. We're not very restricted in who we work with, and we work all throughout the different gamuts and with many different therapeutic areas. Our specialty, where we've really focused, is in an AI-driven tool for fast single-cell image analysis. And so what that means is we're leaders in looking at microscopy images of cells and then being able to pull out lots of different interesting biological signal that would otherwise be quite hard to get to or take a lot of time to get to. And that biological signal ranges from understanding subtly different cell populations, different pathways of cell death, different metabolism dynamics, cell-to-cell interactions, all sorts of things we're very strong at pulling out when looking at cellular images. And our partners tend to use that and the technology we've built to do that to do everything from identifying novel mechanisms, ranking compounds in a screen, differentiating different clinical candidates. They're trying to choose which drug should they take to the clinic, and they want to subtly differentiate the drugs, do responder, non-responder analyses to understand why certain patients are responding to drugs and certain patients aren't, and that sort of thing. But that tends to be our real bread and butter. And you're going to see a presentation from Rachel that walks you through a lot more detail of how we do that. when we work with people, we work on a whole spectrum of different ways of helping groups. And so on the far left side of this slide here, we work, in some cases, we start at the very beginning and we help people do everything from planning their experimental projects, figuring out how imaging and AI and tools can help them with their challenges. We have our own lab where we're very experienced in generating this imaging data specifically for consumption by our AI technologies. We can generate data for you. We do that. We work with lots of different primary samples all the time doing that sort of work. We also sometimes are a pure analysis partner. So folks who have this imaging data and would like help analyzing it, coming to conclusions, that sort of thing, we help them with that. And then on the far right side, what you see here is some companies just license our software themselves and they use the software themselves to try to analyze and draw conclusions and that sort of thing. We are relatively agnostic. We can exist at anywhere along the spectrum, and we just like helping out folks in whatever way helps advance their research goals. So with that, I think I will hand it to Rachel. I'll stop sharing, Rachel, and you can take over. And she's going to take you through a case study in detail, as well as some thoughts on our proposal for how we might be able to help. And please don't hesitate to jump in with questions or if we're off target on the terminology, anything like that.
Yeah, everybody see my screen okay? Okay, sorry, I always get tied up in like the view that Zoom puts all the things, so I can't see everything on my screen. Okay, if you can see my screen, that's the more important part. So yeah, I'm gonna walk through kind of how we generate tooling around the high content imaging data that we generate. So just starting with a schematic of our typical drug discovery workflow here at Spring. So just walking through all the different steps that we typically would take to get from design all the way to drugs. So we do work in high content imaging with cell painting data. So we have a modified cell paint protocol, and I will go through what that means and how we do that. We do take a lot of time at this kind of second step here. So this is a dedicated set of model building and AI training type experiments. This is where we design experiments to get at the biological questions that we're asking. So make sure that we have clear understanding of the data and that the data are optimized for AI analysis. So we spend a lot of time here. We do that in large part because we can do this at small scale, make sure everything's working, and then we do it in a way that we can scale it up into a higher throughput format. So everything that's done here is transferred to a high content screen. This is then generating data that are already built and trainable by the models that we've done over here. So once we've generated our screening data, we apply all the tooling that we've already generated and built. And then that allows us to discover compounds then that shift cells to our control profile of interest. So in the case study that I'm going to share with you, we were looking at human PBMCs, so human immune cells that have been activated in a certain way. And we were looking for compounds that inhibit that activation. So we were looking for compounds that shift from the activated state back to either a basal state or somewhere along the activation pathway. And I'll go into details of that. And then, sorry, I actually like my screen very well here. The last little, I'm gonna minimize this. Okay. The last box here, that circle, I guess it's a circle, is that we can reanalyze these data using other profiles of interest. So when we're talking about different activation states in this particular screen that I'm going to share with you, we can look for compounds that shift cells to different stages of activation. But if we have other control profiles that we've trained over here, we can analyze our screening data to identify compounds that shift to those other profiles as well. So this gives us a lot of data that we can work with and reinterrogate many different ways depending on the questions that we're asking. So the more complicated the biology, as long as we have our controls in place and experiments set up in a way to do this, we can reanalyze the data with new questions.
Hi, can I ask a question?
Yes.
Hi, I'm Jason Seltzer. I'm one of the 8P researchers. I was wondering how your approach differs from the approach of a company like Recursion.
Okay. I might pass this over to Ben or Adi to do the direct comparison because I am not as familiar with what Recursion is doing at the moment. I will say we work primarily in primary cells, and I know Recursion works with cell lines and that they have a very rigid workflow, whereas we are much more flexible and customizable. But I'll pass to Ben or Adi about.
Yeah, so we could probably spend a lot of time on the question, but just to keep it pretty short and simple, it's perfectly reasonable in your mind to think of them as very related approaches. Extremely different business models and companies that have produced different technologies along the way. We are purely focused on enabling others. We're not developing our own drugs, trying to own the IP of the drugs, that sort of thing. Where Recursion is building a full therapeutics company. And so we can partner with all sorts of different folks. We can be very open with our technology. We work with many different groups in lots of different therapeutic areas, whereas Recursion is a more sort of vertical, full therapeutics company. We're an enabling technology for many others. So that's an important thing. Now, because of that, we work with, as Rachel said, many different primary cell types, lots of different disease areas. We have a very flexible set of modified cell painting protocols, whereas Recursion has really sort of narrowed in on some particular protocols and particular data sets that they're mining for their own drug discovery pushes, as opposed to us helping other people with their drug discovery. But it's perfectly reasonable at a high level to think about the thing that we're giving to other partners as related to the thing that Recursion is using internally for themselves as well. There are a lot of differences. There's ways in which I'm opinionated that we're stronger than them, that sort of thing, but I don't think I need to get into that. I think that's a reasonable high level answer. Does that answer your question?
Yeah, thank you very much.
Sure.
Any other questions? While I'm paused. I talk fast and I welcome anyone to jump in. Just interrupt me as I go. Okay, so we use cell painting, as we were just talking about, to capture dynamic cellular features in high content images. So the original cell paints paper released from the Carpenter Lab from the Broad Institute described this method, and it was revolutionary in that it allows scientists to build out morphological profiles of distinct cellular states. So we can look at all of these different features within the cell at the same time and look at the data in a relatively unbiased way and identify patterns in the data and resolve differences that maybe we wouldn't have been able to see with a more targeted approach. These are general cell biological stains and dyes. So these are fluorescent stains that we can use to look at different features of the cell, such as the nucleus. We have all the compartments within the cell, so all the organelles like the endoplasmic reticulum, the nucleoli, which is a subcompartment of the nucleus, and then actin, which is your cytoskeleton, the structure that holds the cell up, Golgi, plasma membrane, and mitochondria, which is my personal favorite organelle. Anyone who knows me probably knows that very well. The painting on the, it's not a painting. This is a microscope image behind me of mitochondria that I took during my graduate work. Not that, anyway. So the whole purpose of this then, though, is to gather as many features as you can from a cell and then analyze those features for structural or cellular states that might be driving different phenotypes or functions in the cell. There is a lot of advantage to this method. It's accessible to most labs because these dyes are widely available and they're relatively inexpensive, and it's scalable. So this is something that can be done using high content imaging. So you can do many, many plates of data. We have modified the cell paint protocol to fit our purposes. We don't stick with the very rigid palette that the Carpenter Lab originally published on. So we also don't combine dyes in a single channel. So we want to be able to differentiate between biologically interpretable features and still get at that unbiased approach of being able to identify patterns within the data set. So to do that, we have separate channels for each compartment that we're staining. We usually stick to 5 to 6 dyes. We are staining human primary cells, which have their own challenges for getting stains and dyes into them. So we've worked out a lot of different protocols for many different cell types. And these palettes, we make them so they're customizable. So this is just kind of a typical palette of what we might use. So looking at nucleus, mitochondria, cytoskeleton, and then we have one stain to stain membranes within the cell. So those typically go around the ER and the Golgi. But we can also add in functional stains, so stains that might give us an idea of cellular health, metabolism, or cell death. We can also do more targeted stains looking for specific protein expression and localization, and we can use those as a ground truth for validating our experimental methods, for validating our models, and to kind of ground us in a direction of this is the path that we're looking for. This has to be true for the cellular state to be defined. And we use our foundational models then to extract features from all of these images that we use for our downstream AI tooling. So we have trained these models to be able to identify all of these little tiny features within the cell to really help quantify what's going on in the cell at any given moment. And then I kind of talked about this already, but I want to spend a little bit more time here. We spend a lot of time making sure that our data are optimized for AI analysis. So this starts with before we even run an experiment, we make sure that we have plate layouts and experimental design that's optimized to measure a biological effect above the biological diversity of primary samples. So what this means is if we're looking for a very profound effect, a very big effect, we might not need as many samples because the variability within the samples is lower. So this is a kind of a classic signal to noise question that we address kind of early on. If we're looking for really subtle differences, we might need more samples to be able to tease that out away from the variability of the samples. So if there's a lot of differences between individuals, but we want to see what's the same about them, we have to kind of look at more samples to get there. But we set up our experiments in a way that we can do that. So we can test 5 to 10 samples. If we can see an effect above the diversity, then we can stop there and train our models. If we need more samples, we've set up our experiments in a way that we can run more plates. We've normalized everything and are able to aggregate those data so that we can compare again and again and again. Does that make sense? I can pause here for a second. Okay. So that's one thing that we set up very early on to minimize any confounders in diversity or variability within the biological data. We also want to minimize experimental variability. This ensures consistent, reproducible results, and it enables us to scale our experiments to high throughput screens. So we use a lot of lab automation to get around experimental variability. I was a bench scientist for a long time. I like to think that I was very consistent every time I pipetted and that I was very consistent and reproduced my experiments exactly as they were done, but I am not a robot. And there is a lot of variability with just using your hands. And so when we use liquid handlers and our lab automation, we're able to minimize that kind of variability and ensure that we have consistent data generated for AI. So AI models identify all sorts of little differences. So as much as we can, we try to keep the differences to the actual biological effect that we're interested in. And lastly, we have a really rigorous quality control assessment of all of our data to identify any variability that's present. So making sure that what we're measuring is what we think we are measuring and identify any confounders. If the stains or dyes are not equally distributed across the plate, if we have a lot of cell death or differences in the confluency of the cells, so the number of cells that we're imaging, those types of things can drive differences that are not the biological effects that we're interested in. So we have a whole process in place here to make sure that we are getting the right kind of data to train and that we understand what we're training. So once we have generated that data, we have this workflow to get to our analytical pipeline. So we have our fluorescent images. We crop these images around a single cell, so literally draw a box around every single cell. We use our models to extract features from those images. So again, when you go back to all the little spots in the cell and the shape of the cell and the size of the cell, we have our models that identify all of those features and they put a number to it. Those numbers are called high dimensional embeddings. So every cell has hundreds or thousands of features that are extracted into these embeddings. And then we can do 2 different processes. These are big buckets. So there's a lot of things wrapped up into this. I'm not going to go through all the details of it here, but happy to address that in a separate call or answer any questions that you guys might have. One set of things that we can do is unsupervised analysis. One of our scientists phrased this as unhuman biased analysis. So this is where you look at patterns in the data, you look for different morphologies, so cell shapes that might cluster together upon specific types of conditions, and you can draw correlations statistically to say this set of cells looks like they all belong in this activated state. This set of cell or basal state, I have it right here, or this set of cells are all associated with our activation condition. We have statistics to show that this is a novel morphology associated with those things, and we can use that to quantify compound effects. We have other ways to do unsupervised analysis kind of along that same metric where we're looking for morphological changes that cluster together to identify different cell populations and biologies of interest. We also can do our supervised modeling. So this is where we would train models based on classes that we assign to them. So if we have a control condition and a test condition, we can say to the model, this is the control set of cells, these are the test set of cells, learn these, and then, hey, here's some cells that you haven't seen before, tell me what they look like. Do they look like my control or do they look like my test? And we can do that at a single cell level, we can do that at a well level. So that basically we can look at like one cell at a time, or we can look at a lot of cells at a time, and really make these classification models ready to identify differences that are driven by compounds. And then we take all of those data that we get, all those tools that we've built for unsupervised cluster analysis, as well as our supervised models, and we can aggregate those into scores that we use to build out cellular profiles. So we're kind of defining what a cell looks like using all of this tooling and saying, this is activated, this is basal. I'll pause here before I get into the case study. Does anyone have any questions? Okay. So all of that I just talked about, we're going to apply now to this case study that we ran at Spring. So this was a phenotypic screen. So this was a screen where we were looking for functional changes. And we were looking for compounds that inhibit a process called inflammasome activation. So inflammasomes, you don't need to memorize this, or really, the only thing you really need to know here is there's multiple pathways that are activated by danger signals in the body. So if you're exposed to a germ or to something very stressful, you get an inflammatory response. And part of that response is through this innate immune pathway or set of pathways called inflammasomes. There's many of these. I have just 3 here. So these little pinwheels in the middle represent 3 of these separate pathways. And those pathways receive that signal and they amplify it. So they tell your body, I need to respond to this pathogen or this danger. And sometimes that gets dysregulated. So sometimes the body starts that process and gets the amplification of that inflammatory response, but doesn't shut it down. And that can result in chronic inflammation or chronic conditions such as gout or rheumatoid arthritis, or more serious conditions that are very dysregulated. This process just keeps going and going and going, even though the pathogen or the danger signal is no longer present. The cells just can't stop receiving some sort of danger signal. And those conditions can result in things like sepsis or ARDS, which is acute respiratory distress syndrome associated with pneumonia. So there's kind of a lot of variability in these different human pathologies. So some are things that a patient would have to take a drug for the rest of their lives, like rheumatoid arthritis, and some are acute conditions where a patient would only need to take a drug, hopefully for a couple of weeks. So up until this point, and kind of throughout, there's a lot of therapeutic focus on these pathways. These are critical for a lot of different human conditions. But most of the focus has only been on one part of one of these pathways. And different indications, because of the differences in presentation, might benefit from different types of inhibition. So something like sepsis or ARDS, where you want to shut everything down very quickly, you might benefit from blocking or inhibiting the process very late in the pathway. So down at the very end stage after these pathways converge. Whereas something like rheumatoid arthritis, again, where someone has to take a drug every day, they might not tolerate something that's like a hammer. So they might want something a little more gentle that's more specific to their indication. So something that blocks very early in the pathway that's actually specific to their condition. So because of that, we set out to identify mechanistically distinct inflammasome inhibitors to be optimally matched to patients with those different types of indications. So we wanted to train models that could identify inhibitors that block very early in a subset of these pathways, to find inhibitors that block kind of in the middle of these pathways, and ones that would block at the very terminal end stage of these pathways. So to do that, we first did our model training set of experiments, where we trained models to identify different stages of activation. So again, we're looking at different stages, as well as across different pathways. So we have models trained to do those things. We then scaled all of those assays to a 12,000 compound screen across 2 activation pathways. So we selected one of these and one of these to run across all 12,000 compounds. We did our modified cell painting assay, where we incorporated into the palette a ground truth stain. So again, this is a stain that helps us validate our experimental conditions, and should always be true for activation and inhibition. So we have a stain in our palette to use there. And we used all of that tooling that we built, applied it to our compound screen, and combined different scores together. So when I say score, we had different readouts from the different types of models, and we put different emphasis on the readouts to identify compounds that block very early in the pathways, compounds that block in the middle of the pathway, and compounds that block at the terminal step. Okay. So I kind of just already said all of this in that last slide, but yeah, we took our images from our screening data. We applied our tooling from classification models. So again, those are the supervised models where we tell the model, this is this thing, this is this thing, tell us the difference. We have different ways to score morphological features. And then our ground truth stain is actually this one down here. And then we used all of those features to differentiate these different stages of activation, to look for compounds that block at those different points. So this is a heat map showing the different scores. So I said we had different ways to score these readouts, and we classified these are just the top 300 compounds from the screen that have been classified either as ATP specific. So we used 2 different activators. ATP is one of them. So we're looking for compounds that block only the ATP pathway. And so those, looking a little more zoomed in on this, are here at the top here. And then on the right, you can see the distribution of our ground truth score across all of the compounds in the library and how they look in the ATP activation state. So we see this right shift over here. So this is showing that the ATP specific compounds have a relatively high ground truth score relative to the rest of the library. So you see that distribution over here. Whereas under flagellin activation, where ATP specific compounds should not do anything, they look exactly like the rest of the library. So they're distributed kind of evenly across with no enrichment for one side or the other, meaning that they don't do anything, which is expected. We see the same but opposite for flagellin specific inhibitors. So these are ones that would block only under flagellin activation. Again, on this ground truth score, we see this distribution shift to the right of the rest of the library for the flagellin state. But under ATP activation, we see an even distribution with the rest of the library, indicating they don't do anything measurable relative to all of the compounds in the library. Convergence inhibitors are ones that would block both ATP and flagellin activation, kind of in the middle of those pathways. So we see a right-hand shift of this distribution of our general ground truth score here relative to the rest of the library. And we see the same with the terminal inhibitors, but we had some other discriminating features for those to see the difference between terminal inhibitors and convergent inhibitors. So all of this to say, we were able to surface many different inhibitors that fall into these profiles that we were interested in, and show that they are in fact mechanistically unique from each other. We then did a downstream orthogonal assay to validate all of these compounds, or not all of these compounds, our top hits. I don't need to walk through all of this unless somebody wants me to walk through each curve here, but essentially what we did was we took 100 compounds that were true hits that scored very well in the screening data. We and validated them in a downstream assay. This was one to make sure that our screening methods could be validated using a different type of experiment. And two, a lot of times if you do screens, you follow up with your hits and they don't behave the same way in a more focused way. So from this top 100 compounds orthogonal assay, 84 of our top hits were validated here. So 84% success of our hits there, which is, in my experience doing drug discovery, is pretty unheard of. And we also were able to do in this orthogonal assay 2 different activation states. These are in primary human monocytes. And we saw that our categorized hits largely performed as predicted from the screen. So we surfaced a lot of really potent, mechanistically distinct inhibitors from this one screen that we could then match to functional alignment between those candidates and indications to help better with patient tolerability and use. So I'll pause here again on the screening data. Does anyone have any questions? Okay.
Sorry. I have a very naive question. Sorry for my voice. I'm a little sick today.
Oh.
So you're utilizing imaging-based systems to look at a bunch of organelles through your painting. And then I'm assuming initially you did some sort of activation to create like a baseline.
Correct. Yes. Yes. So I actually didn't talk through all of the different stages, but yes, we typically, and in this case, we did a set of dedicated experiments to train the models. So we do a kind of smaller scale set of experiments to make sure that our experimental parameters are doing what we think they're doing. We have our ground truth stain. We also had another ground truth measurement. So making these are just established methods to show that we have activation.
Yeah. So let's say you ICTLR3, let's say you take poly-IC, LPS or something and activate your cells, and then you look at activation some way, somehow through staining, right? And then that basically establishes like what an activated state would be, and then you do a drug treatment to see if you alleviate all those things. Got it.
That's right. That's right. And actually, those molecules you just listed are exactly ones that we used. We also used ATP and flagellin. In those initial model training experiments, because we were at a smaller scale, we also did cytokine analysis to look for expression of pro-inflammatory cytokines that get released during this whole process. So we have that as a validation as well, that we were looking at true activation states. And when we train our models, we do not incorporate the ground truth stains. So we do or the ground truth measurements. We train on just the morphological features of the cells, but remove the ground truth stains because we don't want to bias the data in that way.
Got it. And then you're also using PBMC monocytes, which are all floating cells, correct?
Yes.
Which is much easier to quantify, I guess, because you'll be in single cell suspension. Whereas, well, not easier because it's also floating. So for imaging is more complicated. But one of the things that we're interested in, for example, is looking at brain development, and all of the cells that we work with grow in clusters and they're very tight. So how would your system adjust to that, I guess?
Those are great questions. I'll start with so working with PBMCs and monocytes, we've actually worked out protocols to get them to adhere to the plastic somewhat. So we do get enough adhesion that we can wash and stain the cells. So they are stuck to the plate. I actually will pass to Audie to comment on the segmentation of cells that are more confluent and together, but we have methods to do that as well. Audie, are you?
Yeah. So what we have done is, we have trained different models and we have validated those segmentation models, based on human ground truth data. And therefore, based on the type of the data and based on some of the other features, we use different segmentation models, and then we compute the features of each of these segmented cells independently. And sometime, we might, we have this QC tool that may flag certain, fields because the features aren't aligned to the expected range. And those cases we go back and then we add more annotations and we refine the model. So our segmentation is not a one-shot approach, but we basically do it iteratively and we keep refining until we feel it's satisfactory.
And then I'll just add one thing on top of that, which is because a lot of our core technology is doing this AI deep learning based analysis of these images, a lot of the analysis does not rely on segmentation. And that means that we're actually particularly even stronger than other approaches in situations where you have tight clustering, or even when you want to do analysis at a whole field level or at subsets of the field and not at a single cell level. We've done all of those approaches. And so tightly clustering cells are not a problem for us to get that same signal out, even if segmentation gets really hard. Does that make sense at a high level, what I'm saying there?
Yeah, yeah, I understand. Yeah. I mean,
Yeah.
I'll ask you more questions later.
Yeah, I'm happy to go into more detail, and I apologize. I know we're talking about a lot quickly because there's only so much time, but basically our approaches don't rely on having to perfectly segment each cell because we're using modern approaches where you're kind of consuming the whole image and not just the segment of different organelles, that sort of thing. And so it can handle having lots of cells tightly packed in one of those images.
Like, Haru, are you imagining like imaging a neural rosette or something? And like, and how would that be compatible with this?
Yeah, I mean, all of it, right? Anything neuronal based. You start from stem cells, you go into NPCs, you make neurons. All of it is crazy tightly packed. And when you get to neurons, you have axons just floating on top of each other. You know, yeah. Anyway, I understand. It's complicated, but there are ways, I guess.
Yeah,
Have
if
you
it helps.
thought of integrating your system with maybe like expansion microscopy or something like that to basically separate the entire system?
That would be super cool. And Rachel, maybe you want to comment here. We have not done that in the past. That is a kind of brainstormy conversation that has come up in the past, and I don't think anything would stop our system from being able to work in that expansion environment. But I will just say, people fairly commonly come to us in situations where they think they have really complex culture, phenotypic, analytical challenges with various cells. And so what you're describing is not like out of the box, unheard of thing for us to work with you. Yeah. And as Audie's mentioning here, we've worked with neuronal cells in the past.
Yeah, and I'll just add, we're a small team and we are always happy to help in any way we can to modify our methods too, to make sure that we're addressing the questions that you have. And yeah, and we've worked with kind of interesting cell types too. And I think, yeah, I don't see that being an issue here, I guess. So we're happy to work with you. And if you have, we don't have capabilities to build out new microscopy type methods, but if you have images generated elsewhere, we are happy to help analyze those too. Any other questions here? Okay, I'll just hop in. We have a few minutes left to talk through what our plan could be for 8P Heroes. So again, just starting with this drug discovery workflow that we have at Spring, we could start with the cell painting, high content imaging side of things. So we have customizable palettes that we could address for hero specific biology. So if there's specific stains or subcellular features that are associated with 8P, then we can incorporate those into our palettes. So we can add in a lysosomal stain, for example. We have mitochondria, endoplasmic reticulum, and we can make sure that those stains are robustly working in our palette. And we would take careful care to make sure that we would carefully design the experiments to highlight what makes 8P Heroes unique. So what is the biology that is driving their characteristics and how we can actually identify those biologies in the early stages of experimental design. We could then spend, and I think this would we've separated this out as to an AIM-1 for model training experiments. So this is, again, that smaller scale that we do to validate experimental protocols and methods, and all of our data generation and feeding into models that are learning these specific profiles of heroes and their controls. And in this process, we would also work to do discovering of cellular subtypes. So using that unsupervised analysis to identify cell morphologies or cell shapes that are associated with either the hero samples or the control samples. Once this AIM-1 would be completed, once we're confident in the tools that we've generated here, we could scale all of that into a phenotypic compound screen. So phenotypic screening, again, is where we're looking at functional differences. So we're not doing a targeted approach. We're looking for compounds that could drive a shift from one state to another. And we can scale all of the tooling and experiments that we've worked out here into the screen, and where we would treat hero cells with an annotated library of known bioactive compounds. So these are compounds that have already gone into humans that are generally regarded as non-toxic and relatively safe. Again, we'd have to work through all of what comes out in the end to make sure that that's the case still, but we have this library of bioactive compounds. Then we would generate those data and apply all of the tooling that we did in AIM-1 to the screening data to discover compounds that shift cells from the hero states to a control state. This would give us hope, and we can create profiles along a spectrum here as well, looking for compounds that could help as therapeutics. And again, we have a lot of focus on setting up our screens and setting up our plates in a way that we can reanalyze data using multiple profiles of interest. There's a lot of individuality involved here that we would certainly try to incorporate to make sure that we're.
And what is the cell type that's being proposed to evaluate here?
Yeah, so I think it was fibroblasts. Was that correct, Ethan? I think we were, yeah. And we have worked extensively with fibroblasts in the past as well. So that's a cell type that we have run screens on and have a lot of. I'm sorry, I couldn't hear.
No, just a temporary hot mic.
Okay. Yeah, so this is kind of how we would propose. We're all, of course, happy to customize and work with you guys, but we're confident that we could transfer our knowledge and then our expertise to help run this kind of a screen and look for therapeutics.
So I guess the question in my mind, maybe others, is, what is the relevance of the human PBMCs in this type of a disease? And not that we don't think it's a multi-system disease, we just sort of have had a neurological focus with disease modeling. But I guess, because you're starting out with the fibroblasts as your initial input, like, I guess intuitively I would have thought you would just do the screen on the fibroblasts, not the PBMC.
Yes.
Is that like swappable?
Yeah. Oh, sorry. I did not correct that. That is my fault. This should be cells or fibroblasts. Sorry, this would not be done on human PBMCs.
Okay.
A typo, or not a typo, is an unfixed thing on my part. Sorry. The screen would be done on the same cells that we've trained models on. So fibroblasts here and here. Sorry about that.
Yeah, no, that makes sense.
Yeah, makes more sense than shifting to PBMC. Sorry.
I guess, did anyone else have any thoughts? I know we're winding down, but I wanted to maybe use this as a little mini brainstorm where, I think the idea is pretty pressure tested, but if anyone else has any thoughts, especially on, I've seen a cell painting screen done on fibroblasts. I've been part of that project for another client, and it's actually worked out kind of better than I would have expected, honestly. So I believe in the thesis that you want to normalize the feature set and look, make the disease cell look more like an unaffected cell. But I guess, if anyone else had any higher level questions or higher level concerns, please use this moment now.
Hi, one question that I had was whether the system could be used for genetic perturbations. If you imagine an 8P hero who has a large duplication on 8P, I could imagine, knocking down each gene on 8P and then seeing which one pushes the phenotype closer to the control. And that could be a way to identify the dosage sensitive drivers if your system is compatible with, high throughput CRISPR or RNAi.
Yeah, yes, we could definitely work on something to that effect to do genetic perturbations. Yes. We have a lot of experience in liquid handlers and handling different types of perturbations. So yes, I think we could make that work. Yeah.
Maybe a naive question, but so you start from, just to give a sense of the granularity maybe of the data, you start from 5, 6 different staining. And then I'm wondering how many on average, or maybe it's very variable depending on the setting, molecular morphological features you usually extract, and who decides how subtle is the difference and what is meaningful?
I'll take a stab at this, and then I think Audie's probably a better person to answer this. So yeah, we start with 5 to 6 channels, typically looking at all the different features that we can, and the actual features that are used by the models are, my understanding is they're pretty granular. Audie, actually, I'm going to pass this to you now for that question.
Yeah. So what we do is we compute, both the AI-based features, your embeddings, as well as your segmentation-based features independently for all the stains. And then we figure out, which are, and then we also do the combinations. In fact, our platform allows you to combine any set. Like, for example, you only want to use, let's say, nuclei-based stain and see, what kind of mileage you get. But sometimes you might want to add something else. So we allow you that flexibility, and then for each of the feature, you have this combination, and sometimes we say there is an expensive stain which is really useful, but you don't want to replicate that. Then we use some of our supervised method to say, can we use the existing stains in combination to try to give you a feature that correlates well with expensive stain that you don't want to add every time.
Barbara,
At the end of the day, like, because I think from the other project, the PAX2 project, I mean, if I was remembering the data right, it's like you end up with maybe even a couple of hundred features, right? And a feature could be the size of this, Golgi stain speckle in relation to the nucleus, or however you want to kind of parameterize it. But it seemed like you're talking about potentially hundreds of features, right? Is that the order of magnitude?
that is, but we're flirting with a really important point that we probably don't have the time to go into all the detail of, but it's really important, and it's part of what differentiates us from these other approaches that you've seen, which is, if we were giving you a deeper technical walkthrough or a technical demo of how our system works, we do 2 different things. One is we generate what we'd call traditional features, and those are the things, Ethan, that you're referring to, where we can describe it to each other. It has something to do with the shape of the cell, radius, relationship between this organelle and that organelle. It's something that we have words to describe what's going on in the cell, and we generate hundreds of those, and we can use those. However, a place where Spring really shines and focuses is we do this embeddings-driven analysis, which is letting the AI quantify all the things it thinks are interesting in these cells, which gets into some of those things are hard to describe to a human. These are very complex sets of biology and relationships. But we've done extensive benchmarking and work in many different scenarios where you're trying to predict MOA, you're trying to call hits using standard industry benchmarks and benchmarks we've created. And that approach of letting the AI pull out tons, thousands of features that are hard to describe to a human consistently outperforms this other method that's more classical in the industry. And there's only a few companies, like we do this, Recursion does this, but it's internal to Recursion. This is where, if you're willing to, as you said, Ethan, accept that you want to make a hero cell look more like this control cell, things like that, that technology consistently outperforms the features where I'd be able to say, we have 100 or 200 of these human describable features. And I realize I'm talking about sort of maybe frustratingly subtle points, but it's
No,
important.
I think
because
actually
of it.
I
Does that
got
make
it.
sense?
Yeah, now it makes 100% sense, right? So hopefully that also landed for everybody else. I know we've got about a minute left here. I think, this is not the final conversation here. We just wanted to offer you the stage here, the community stage, but we'll definitely have time for more follow-up. I guess any concluding thoughts from the audience, or I think this was a pretty solid session. So really appreciate all the firepower, brought by the Spring team, and shows their commitment.
Yeah, I mean, if I could just say one thing, which is obviously we're really excited to work with you as a company. The excitement goes beyond, and you have a bunch of individuals on this call who really are motivated by what you do. And even if it's separate from a partnership, if you want to get on the phone with Rachel, Audie, me, brainstorm, have discussions, you've got a bunch of people who just want to help. And so feel free to use us in that regard too, as well as the more formal, can we work with you and help your research type of thing. But we would love to help in some way.
Great. No, definitely appreciate the open door. Parting words of wisdom, Bino. Let's wrap this up.
Oh, yeah, I wish I had some. Thank you. I'm really grateful for you guys and just being open and willing to help us. We're a certainly complicated and rare chromosomal disorder. And so, thanks to our scientists too, to just kind of pitch in here and think about how we can get some at least early prelim results on cell painting. I think it's a gap. Hopefully there's some insights generated from that. So again, we fully understand kind of weighing what we're going to actually get out of it and are realistic about it. Again, we'll see you all very soon on the next roundtable.
Thanks, everybody.
Thanks all.
Thank you.
Have a good rest of your day.
Take care,
Thanks.
everyone.